Welcome back, my dear students. Today we are going to take a closer look at the structure or structural organization of skeletal muscle, the amazing framework that makes movement, posture and strength possibly. Muscles may look simple from the outside, but inside they are built with a highly organized arrangement of fibers and tissues. Each muscle is wrapped in a connective tissue covering, packed with bundles of muscle fibers and lined by the sarcolemma. Within these fibers lie myofibrils, which are further divided into repeating units called sarcomeres, the true engines of contraction. Inside the sarcomeres, special proteins like actin and myosin interact to generate the force that drives all muscle activity. By the end of our discussion, you will have a clear picture of how muscles are structured, right from the outer covering to the microscopic contractile machinery that keeps our body in motion. So, the whole muscle, which is connected with the bone or our skeletal system, which is called as the skeletal muscle. The whole skeletal muscle is wrapped in epimysium. This is epimysium. This is the dense irregular connective tissue, and within whole muscle there are so many muscle fascicles. They are wrapped in perimysium. This is muscle fascicle and they are wrapped in perimysium, and within each muscle fascicle there are so many muscle fibers, which are also known as muscle cell or muscle myocyte. They are wrapped in endomysium. This is covered by endomysium is actually the areolar connective tissue which covers the cell membrane. So just beneath the endomysium there is cell membrane, which is also called as sarcolemma. So, if we consider the myofibril, this is the unit of the muscle fiber or the muscle cell. This is the myofibril. It acts as a train. You can consider it as a train. So, the different this is the myofibril, and so the different repetitive unit of the myofibrils are called as the sarcomere, which is also the contractile unit, which is the unit of contraction of the skeletal muscle. This can be comparable with the train car or the each compartment of a train. So this is the organized collection of myofilaments. Myofilaments are made up of actin and myosin protein, which are the contractile proteins. These are the thin, the pink are the thin actin protein and the blue are the thick myosin protein. So this is the whole muscle and covered by epimysium. This is the muscle fascicle covered by perimysium, and this is the myofibrils covered by endomysium, which is also known as muscle cell. It has sarcolemma, and another sarcolemma is also called as cell membrane. Inside the endomysium there is sarcolemma. So the myofibrils is just like train, and the sarcomere is the train car or the compartment of a myofibril. Their sarcomere is basically the organized group of myofilaments. They are made up of actin and myosin. So myofibrils are greater and myofilaments are the protein component of the myofibril. So myo means muscle and cyte means cells. So myocyte means muscle cell. So AP is the outer layer, perimysium is the middle layer, and endomysium is the inner layer, and just beneath the endomysium there is the sarcolemma, the cell membrane of the muscle cell or myocyte. So what is the ultrastructure of skeletal muscle? Basically, skeletal muscle is an organ made up of hundreds to thousands of long muscle fibers or myocytes or muscle cells, and each fiber is an elongated and contains many nuclei. Muscles also have connective tissue, blood vessels and nerves to support their function. Connective tissue and fascia, subcutaneous layer, hypodermis, this is made up of areolar and fat tissue. It separates muscle from skin, it stores fat, it insulates and protects. Fascia, it's a dense connective tissue sheet around muscle. It helps free movement and carries blood vessel, nerve and lymphatics. There are three layers of connective tissue inside muscles. Epimysium is the outer layer, surrounds the whole muscle. Perimysium surrounds bundles of 10 to 100 plus fibers called the fascicles, and endomysium surrounds each individual muscle fiber made up of fine reticular fibers. These layers combine to form tendons. Tendons are basically rope-like structures attaching the muscles to the bone. What is aponeurosis? Aponeurosis is a broad flat sheets, which is the example epicranial aponeurosis. So we need to know the nerves and blood supply of the muscle cell. Somatic motor neurons control the skeletal muscle, and each action branches with every branch supplying a different muscle fiber. Capillaries surround each muscle fiber, supplying oxygen and nutrients and removing waste. Since muscles need a lot of ATP for contraction, blood provides oxygen, glucose and fatty acids. This is the image showing the skeletal muscle. This is the muscle fiber bundle. This is the muscle fiber. It consists of myofibril, and these are the myofilaments made up of actin and myosin protein. Actin is the thin filament or made of I band, and A band is the made up of thick filament or myosin protein. This is another I band. In between two I band there is an A band. I band is basically isotropic band, and A band is anisotropic band. So there are zigzag lines which are also known as jet disc, and this is the sarcomere, the alternative repetitions of the thick filament and thin filament. Thin filament is made up of actin protein and thick filament is made up of myosin protein. And just in the middle of the sarcomere there is a line known as M line, and within the M line there is a H zone. So muscle fiber structure. Muscle fiber is basically 10 to 100 micrometer diameter and length is 10 to 30 centimeter. Each fiber is originated formed by fusion of many myoblasts, explains the multiple nuclei. Number of fibers is fixed at birth. They cannot divide, they only grow in size. Important parts of muscle fiber: sarcolemma, this is the plasma membrane of the fiber. T-tubules are the transverse tubules. They are small inward folds of the sarcolemma that carry nerve impulses deep into the fiber. Sarcoplasm or cytoplasm contains glycogen, energy store for ATP, myoglobin, oxygen binding pigment, and mitochondria near contractile proteins for quick ATP supply. This is the structure of the muscle. This is the sarcomere, the repetition of the myosin and actin protein, and in between there is H zone, and within the H zone there is M line. Myofibrils and sarcoplasmic reticulum. Myofibrils are long, thin, contractile structures. They are 2 micrometer wide. They are running the whole fiber length, give the striated look. Sarcoplasmic reticulum is a network around myofibrils that stores calcium ions needed for contraction. Terminal cisterna. They are enlarged sarcoplasmic reticulum beside T-tubules, and the triad is the 1 T-tubule plus 2 terminal cisterna, which are essential for muscle contraction. Thin filaments, which is 8 nanometer wide, mainly made up of actin, and thick filaments are 16 nanometer wide, mainly myosin. Arrangement: each thick filament is surrounded by 2 thin filaments, and these are organized into repeating units called sarcomeres. So sarcomere is positioned between the 2 successive jet discs. In between 2 jet discs there are sarcomere. Bands and zones of a sarcomere: A band, this is the thick and overlapping thin filaments. I band is the thin filaments only, I is equal to thin, and H zone, this is the thick filaments only, H means heavy or thick. Jet disc anchor the thin filaments. M line holds thick filaments in the out center. The pattern of these bands give muscle its striated appearance. So the triad is made up of T-tubule, terminal cisterna of the sarcoplasmic reticulum, 1 T-tubule and 2 terminal cisterna. Types of proteins in myofibrils: there are contractile proteins which can generate force, like myosin, which are the thick filament motor protein with a golf club head. This is the myosin protein. And actin is the thin filament, it has binding sites for myosin. Regulatory protein which control the process of contraction. In next class we will learn the mechanical and chemical basis of muscle contraction. So tropomyosin blocks myosin binding sites when the muscle is relaxed, and troponin acts as a calcium sensor, moves tropomyosin so contraction can occur. So the other structural proteins which provide alignment, elasticity and stability of the muscle cell, they are titin. Their elastic protein helps muscle return to resting length. Alpha actinin, it anchors thin filament to the jet disc. Myomesin holds thick filament at the M line. Nebulin, it helps align thin filaments and controls their length. Dystrophin links thin filaments to the sarcomere, transmitting force to the tendon. In today's class, we discovered that the structures of muscles is beautifully organized from the outer connective covering down to the microscopic contractile units. Each muscle is made up of bundles of fibers wrapped by protective layers and lined by the sarcolemma inside. Sarcolemma is the contractile unit of the muscle cell. Inside myofibrils, they are arranged into repeating sarcomeres where proteins like actin and myosin interact to produce contraction. This precise arrangement is what makes muscle strong, flexible and efficient. So always remember, the strength of our movement lies in detailed structure of our muscle. So stay curious, keep learning and see you in the next class. Thank you.